I want to do a review of the physiology of potassium homeostasis, and then following that, I'll give a presentation on hyper and hypokalemia. Potassium moves into our body from ingestion. It goes into our GI system and it moves into the portal blood circulation. The portal blood circulation typically goes solely to the liver, but some of it gets moved into the pancreas, and the pancreas is the first organ to realize that there is an increase in potassium. Potassium then moves into the beta cells and causes the excretion of insulin. This is important because insulin then drives the potassium that you just ate into the other cells in your body. With that said, let's quickly review how insulin is produced and excreted from the pancreas. You'll remember that the beta cells in the pancreas, and this is a beta cell, the beta cells have constitutively active GLUT2 transporters. GLUT2 transporters are those transporters that can transport glucose into a cell without insulin activity. So here's my glucose transporter, the GLUT2. Here's my insulin in secretory vesicles. This is my sodium potassium ATPase. This is the ATP-dependent potassium channel. And here's the voltage-gated calcium channel. So typically, without the glucose load from food, like in our fasting state, the beta cell will be sitting there maintaining a membrane potential of negative 70 millivolts, and it does that with the ATP pumps. These pumps drive in 2 potassium and drive out 3 sodium and maintain this membrane potential. And now, the only ion that creates a membrane potential is the one that has the most permeability. So potassium that moves in, it has to be able to move back out freely in order to reach its equilibrium potential. So it's moving out through this potassium channel, this ATP inactivated channel. Whenever we eat and we get a glucose load, that glucose can move in freely through the GLUT2 transporter. It's then metabolized into ATP, and it gets a whole excess of ATP. That ATP will then inhibit this potassium channel. The potassium level builds up and the membrane potential moves from minus 70 to minus 50. And just as I've written here, minus 50 is the threshold for opening this voltage-dependent calcium channel. The calcium channel opens up, calcium moves in, and it stimulates the exocytosis of insulin. And that's how the insulin gets out of the beta cell. What I'd like you to notice is that there were 2 things that were required. There were 2 things. The 2 things were glucose and potassium. So when you eat that potassium, it plays a part in the excretion of insulin. If you were to eat a load of glucose with no potassium, you'll still get insulin secretion. But when you eat a food bolus that has lots of potassium in it, it enhances the insulin secretion. So then what does insulin do? First, it's going to bind to its receptor, and by binding to its receptor, it's going to allow this GLUT4 transporter to be moved to the surface. GLUT4, by the way, is the glucose transporter that's found primarily in adipose tissue and primarily in skeletal muscle. So it moves that to the surface, and now glucose can move in. So now that glucose has moved in, it can go on to make ATP. That ATP then can power the sodium-potassium ATPase. That doesn't mean that the sodium-potassium ATPase hasn't been powered all along. It just means that it's getting extra power now. It's not running on low gas. And so the potassium moves into the cell. This is great for the immediate high load of potassium. But if this was all that happened, the cells in our body would just get chock full of potassium, and we just keep getting more and more and more. And you know that's not the case. So the principle of physiologic homeostasis is that the intake of something is equal to the output of that same thing. So whatever potassium we take into our body, it has to equal the potassium we excrete out of our body. And that's where the kidneys come in. So in general, all of the potassium that is absorbed in the GI system each day will be exactly excreted by the kidneys that same day. That is to say that GI absorption will equal renal excretion. And so a quick review of the renal handling of potassium is we see that in the proximal collecting tubule, the PCT, that we have potassium reabsorbed. So it gets filtered, all of the blood and potassium gets filtered here, and that potassium gets reabsorbed in the proximal convoluted tubule. And then you see that it gets absorbed right here in the thick ascending limb of the loop of Henle, and then over here again at the distal at the far end of the distal convoluted tubule and the collecting duct. In general, 67% of potassium is reabsorbed here, and it's done isoosmotically, so the exact same potassium and the exact same amount of water, all of that stays isoosmotic. 20% is reabsorbed in the thick ascending limb, and then the rest, that is 87%. 87%, almost exactly those amounts, are reabsorbed in those two places, and so all of the fine tuning is done right here at the collecting duct. So in general, there is a 13% play where we can excrete up to 13% of the filtered load, or down to as low as about 1% of the filtered load. And this number can actually go up because we know we can alter the physiology at the thick ascending limb of the loop of Henle, things like loop diuretics. And so you could potentially get this up to 20 to 25%. But in general, under normal physiology, you have between a 1 and 13% range that you can alter renal potassium handling. And almost all of that alteration is done in the principal cell. A tiny, tiny bit is in the alpha intercalated cell. So let's take a quick look at how the principal cell works. The principal cell has these sodium channels called ENaC channels, so sodium can freely move in, and then sodium gets pumped out the basolateral membrane by the sodium-potassium pump. That creates a concentration gradient, pulling more sodium in. The sodium-potassium pump pulls potassium into the cell, and the potassium can either diffuse back out the basolateral side or it can diffuse out into the lumen. Whether or not it diffuses into the lumen is all determined by the electrochemical gradient, also called the electrochemical lumen potential. So really quickly, things that can alter that. The things that can alter the lumen potential include high sodium delivery to the collecting system, high or low flow rates. So high flow rate would cause, and basically what that's doing is, if I pump a potassium molecule out here and it just sits here, then I don't have, and I want to pump out another one wants to diffuse out, well that one's still sitting here. But if I have a high flow rate, that gets washed away really quickly, and so that now there's room for another potassium to get, to diffuse out. With the high sodium delivery, basically what that does, sodium diffuses in freely, a high amount a lot will diffuse in, and this sodium-potassium ATPase will pump a lot of potassium in here. Lots and lots and lots, and that drives up the electrochemical potential from the inside rather than from the outside. Another thing that will do it will be anionic molecules, something like organic anions and various anions that get delivered to the distal collecting duct, such as bicarb, such as certain amino acids. Now, most amino acids are reabsorbed in the proximal convoluted tubule, but under some pathologic circumstances, they can get delivered to the collecting system. But the point I'm making here is any negatively charged particle that is stuck in the lumen can change the electrochemical potential. Now, sodium moving in does not create a positive outward force because it usually is absorbed with chloride anions. Sodium and chloride get absorbed together. And finally, aldosterone has an impact on the electrochemical potential. Aldosterone binds to, so it moves freely across the membrane because it's a steroid hormone, and it binds to the intracellular mineralocorticoid receptor. The mineralocorticoid receptor then upregulates the production of the ENaC channel and the basolateral sodium-potassium ATPase. And to some extent, it also upregulates the luminal potassium channels. So in this way, it not only changes the electrochemical potential, but it also can change the permeability, favoring potassium to be excreted. I wouldn't be doing complete justice if I didn't go over the alpha intercalated cells. This cell is important especially in a low potassium diet. And what happens is you have a low potassium diet where you're barely getting enough potassium, almost zero potassium, then these cells are what get you the maximum bang for your buck. Otherwise, these cells are not important. So super low potassium diet, you are acidifying the urine in exchange for absorption of potassium, and that potassium is pulled into the basolateral side via a potassium chloride cotransporter. You can get a modest alkalosis from this, but it's only active at the extreme lows of potassium ingestion. And in some situations, insulin will also be very low. And with insulin very low, you'll also get some intracellular potassium shift to the outside of the cell to maintain a somewhat normal potassium without this mechanism at all. So I mention it here for completeness sake, but it really isn't extremely important. What is important is the principal cell and the effect of aldosterone. And since aldosterone is important, we have to understand what things can inhibit or upregulate aldosterone in order to understand what can be involved in the pathophysiology of potassium homeostasis. So here I have a graph of the glomerulus, and the important part of this is you can see, first of all, that right here blood is coming in. So this is the afferent arterial that I'm coloring in right here, and then this is the efferent coming on its way out. And at this afferent arterial, I have this special group of cells. So it starts right here. I'm highlighting this group of cells. This is called the juxtaglomerular apparatus. And just so I can make sure you see it, I've highlighted these cells right here. And this part of the juxtaglomerular apparatus is called the macula densa, and this is the granular cells, and these are the mesangial cells. And you'll notice that the mesangial cells go into the glomerulus, but there are some that are outside the glomerulus. The mesangial cells that are outside of the glomerulus contribute to this juxtaglomerular apparatus. And this unit works together in three different ways. First of all, you can see, of course, these granular cells, like for right here, for example, and these ones I'm coloring them all in, these granular cells, they have a location right next to the afferent arteriole. So because they're right next to the afferent arteriole, they get information about how much the afferent arteriole is being stretched or contracted. They can sense how much pressure or volume is moving in, and so they are baroreceptors. So if there's low flow rate, your body says, hey, I'm low on water, I want to reabsorb some more water, I want to save my water. And so that's one way that it senses low water, is by being a baroreceptor. Another way is, suppose this is my glomerulus, my proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. Suppose if my flow rate is really high coming in here, then by the time I get past the proximal convoluted tubule, I'm not going to have reabsorbed all of my sodium. I'm going to have a lot of sodium that I just didn't have enough time to reabsorb because the flow rate was too high. When it gets back up here to this thick ascending limb of the loop of Henle, the sodium potassium 2-chloride pump is going to pump a whole lot of sodium into the intracellular space, and that intracellular space is going to communicate to the juxtaglomerular apparatus that you have a high flow rate because I'm seeing a lot of sodium here. And a third way your kidneys are going to be able to sense how much volume you have. So their job is really, I want to make sure I have enough volume to maintain blood pressure. And that's everything that it's doing down here. If you get a low amount of sodium coming here, it's saying, oh, I filtered really low volume and I was able to get all of the sodium reabsorbed right here, because whenever you get back around to this thick ascending limb, you don't have any sodium there. So it's going to say, oh, low volume, I need to reabsorb more water. Or if you are coming into the afferent arteriole with a low pressure, those baroreceptors are going to say, oh, low volume, I need to reabsorb more water. And the third way is, if you have a low pressure coming out of your heart, there are baroreceptors on the large arteries that are going to signal the release of adrenaline and norepinephrine. So epinephrine, norepinephrine, those are going to go back and activate the beta-1 receptors in the juxtaglomerular apparatus. All three of those things are going to cause the secretion of renin. So that's what this is all about. We have a renin-angiotensin-aldosterone axis, and the way that that axis behaves is, first, renin gets secreted by juxtaglomerular apparatus, and it gets secreted in response to three things: decreased renal blood flow, low sodium delivery to the distal tubule, and beta-1 adrenergic receptors. Now, renin is also directly stimulated by some prostaglandins, and it is inhibited by other prostaglandins. Out of all these things that we've talked about, the one that is really, really interesting and worth looking further into is this mechanism that's in the distal tubule. It's in I say the distal tubule here, this is more of a typo. It's more the thick ascending limb of the loop of Henle and maybe the early distal tubule. So this is a cell in the thick ascending limb of the loop of Henle, or the distal convoluted tubule. It's mostly in the thick ascending limb. So this has something called a sodium-potassium 2-chloride cotransporter. And it's usually written as NAK2Cl cotransporter. And this uses the sodium concentration gradient to drive sodium into this cell in the thick ascending limb. And then, of course, a sodium-potassium ATPase to drive sodium out the other side. So sodium's moving down its concentration gradient, pulling these other cations and anions in with it. It gets in here, and the sodium-potassium ATPase pulls sodium out the basolateral. At this point, there's two possibilities. Either this sodium-potassium ATPase pump is working hard enough and no sodium builds up in here, or it's not working hard enough and we get a lot of sodium building up inside the cell. If the sodium does not build up, a complex signaling mechanism leads to the production of prostaglandin E2 within the macula densa. And I'm going to zoom in really quick to show you the macula densa again. So this was the macula densa right here. So as long as there is no sodium building up, that means that the sodium concentration being delivered here is relatively low, or in other words, that the blood flow to the kidney is low, and so all of the sodium gets reabsorbed early in the renal tubule, and sodium delivery here is low, then it will cause prostaglandin E2 to be made. And that prostaglandin E2 can stimulate renin release in the juxtaglomerular apparatus. On the other hand, if there's a lot of sodium builds up, that's telling the kidney, hey, the flow rate is way too high. And it's going to result in water being pulled in and cellular swelling. This activates a stretch-activated maxi-anion channel. This anion channel allows ATP to move out. ATP is activated on by ecto 5' nucleotidase, which converts it to adenosine. Now, I'll just remind you, adenosine is a methylxanthine. There are two other molecules that are very common and known to be in the methylxanthine class. One is caffeine and one is theophylline. So caffeine can act to block the activity of adenosine here. There is still, regardless of the fact that we have shown it to be true in test tubes, there is still some controversy over whether or not caffeine does act like this in the kidney. But just for argument's sake right now, we'll assume that caffeine can block whatever activity adenosine is about to have. So adenosine will bind with high affinity to the A1 receptor and lower affinity to the A2 receptor. So the primary receptor, the adenosine 1 receptor, causes vasoconstriction of the afferent arteriole, and it also inhibits renin. It's saying, hey, there's too much blood volume. I don't want to activate renin because I know doing so will increase the blood volume, so it inhibits renin, and it vasoconstricts the afferent arteriole because it's saying, hey, there's hyperfiltration here. There's too much water getting filtered in here. I need to constrict this down. And so most of what the evidence shows is that at maximum affinity of this mechanism, we can get a reduction in GFR by about 20%. So if, for example, if GFR is 20% of renal blood flow or renal plasma flow, then reducing that by 20% would mean it would go down to 16% of renal plasma flow. So if sodium doesn't build up, like I already said, a very complex mechanism leads to the production of prostaglandin E2 within the macula densa. It can bind to the EP2 and EP4 receptors to stimulate renin release. It also causes vasodilation of the afferent arteriole. This will cause an increase in renal plasma flow and also an increase in the glomerular filtration. So we went through a bunch of slides already, and we're still at like, so what for potassium, right? So we have this low delivery of sodium. We have an increase of prostaglandins. These prostaglandins stimulate the secretion of renin. And remember, if you're taking an NSAID, NSAIDs can blunt this effect. They can stop the production of prostaglandins via cyclooxygenase. And so what does prostaglandin E2 do in the kidney? It stimulates renin release and it dilates the afferent arteriole, and that can both be blocked by the use of NSAIDs. NSAIDs will cause a decrease in renal blood flow. And so now we're asking, so what? How does this lead to aldosterone? So here's the schematic. We have a low blood pressure. That's what it's all about. This whole axis is about keeping the blood pressure, the mean arterial pressure, at a nice constant state. And so that means there's decreased renal perfusion and there's a low pressure in the carotid and aortic baroreceptors. You get a low pressure in the afferent arteriole and a decrease of sodium delivery to the thick ascending limb because of this renal hypoperfusion. And then you get a high sympathetic tone because of this baroreceptor activation. So all three of these things lead to renin release. Now, the decreased sodium leads to prostaglandin E2, which leads to increased renin release. And all standing over here by himself, already taking place. He doesn't care. The liver doesn't care what the rest of the body's doing. He's been making this thing called angiotensinogen and secreting it out into the blood, into the plasma in the blood his entire life. He's just constantly making as much as he can, not as much as he can, but making a lot and sending it out. And it's just an inactive molecule until you get renin acting on it. When renin acts on this guy, it converts it into an active molecule. So renin cleaves angiotensinogen into angiotensin 1. And then in the lungs, angiotensin converting enzyme converts angiotensin 1 into angiotensin 2. And I just want to take a side step and remind you that prostaglandin E2 has dilated the afferent arteriole. Now back to angiotensin 2. He causes vasoconstriction, systemic vasoconstriction, which will lower the blood pressure as a negative feedback mechanism. He also constricts the renal vasculature, but primarily the efferent vasoconstriction. So if you look at the afferent arteriole, prostaglandin E2 is causing dilation. The efferent arteriole, angiotensin 2, is causing vasoconstriction. This has a net effect of maintaining renal blood flow and increasing glomerular filtration rate. So our GFR gets back to normal. Angiotensin 2 also goes into the adrenal cortex and causes the secretion and production of aldosterone. Aldosterone is going to act primarily by causing sodium reabsorption, which secondarily causes water reabsorption. But in the process of doing that, we're going to lose potassium and hydrogen. So this is what one mechanism. There are three mechanisms involved in contraction alkalosis. This is one mechanism of contraction alkalosis. That is that at low volume states, the aldosterone that is secreted causes us to lose hydrogen ions into the urine, making the blood plasma more alkalotic. So if you are to find a patient and find hypokalemia and a very high pH, then you would first, just that pattern should say contraction alkalosis. Now this is about potassium balance. So not only can a renin-angiotensin axis activate aldosterone, but hyperkalemia can activate aldosterone by an angiotensin 2 independent mechanism, which ultimately results in potassium secretion. So that water retention is going to feed back and inhibit. We've got vasoconstriction that feed back and inhibit. Then also this increased sodium absorption will ultimately come back and inhibit because we have an increased GFR. This increased GFR will feed back and inhibit. And hopefully after we drink a cup of water, this whole thing will shut down. And so that's a quick look at potassium homeostasis. There are two ways of looking at potassium homeostasis. There's the internal potassium balance and there's the external potassium balance. So internal is saying that there's a balance between this intracellular space, this intracellular fluid, and this extracellular fluid. It's a very, very delicate balance. And external potassium balance is saying that anything I consume, GI tract, is going to be excreted renal. And ultimately our formula, our mass balance should be that renal excretion is going to equal whatever I consume plus any potassium lost from dead cells minus any potassium uptaken for the production of new cells. And already you can kind of get an idea of what might cause hyper and hypokalemia. For example, if I have an inhibition in the renal excretion of potassium for some reason, call it renin problems, call it aldosterone problems, call it I don't have enough GFR, low volume problems. If I have a change in consumption, like I am just anorexic completely, I'm not getting enough consumption, I still have an obligatory renal excretion. Another one, you see cell lysis. If I have a high turnover of cells, or if my turnover of cells for some reason falls while my new cell production rises, I could pull too much potassium out of the extracellular fluid and cause a hypokalemia. And so that is the external balance. The internal balance would say if I had some problem maintaining this membrane shift right here, this transmembrane potassium shift, then I could either get hypo or hyperkalemia. I'm going to wrap up with this slide for the physiology of potassium homeostasis. So the internal potassium balance is mediated by primarily one transporter, the sodium potassium ATPase transporter. So 98% of your whole body potassium is kept inside your cells by these pumps. The other 2% is in the extracellular fluid. So I read this and I said to myself, I want to do a sanity check. I know that the intracellular potassium concentration is roughly 150 milliequivalents per liter, and the extracellular potassium is approximately 4.5 milliequivalents per liter. And so I was thinking, well, how much mass? Because it's saying that 92% of the potassium mass is inside the cell, 2% of the potassium mass is outside the cell. So mass, you'll remember, is the concentration, which we've got the concentration in both places. Concentration is equal mass over volume. So I can rewrite this formula and say that the mass is equal to the concentration times the total volume. So intracellular fluid, your body, on average, everyone has about 23 liters, and extracellular fluid, on average, 19 liters. And so 150 times 23, I've got 3,450 milliequivalents of intracellular potassium and 85.5 milliequivalents of extracellular potassium. Now, if I were to add these together to get the total potassium in my body, 3535.5, and I divide that by the extracellular, I've got 2.4%. So my sanity check says that, hey, I've said that I should have 2%. I'm calculating 2% from my standard numbers that I know. I got a little bit curious and I said, well, I want to know how much total body potassium I have. And so one thing I did is I said, okay, these are milliequivalents, 3450 milliequivalents. And I know that potassium has an atomic mass of like right around 39. I'm just going to say 39.5 because I don't remember the exact number. Whenever you multiply this all out, you get like 100, I think it was 135 or 136 grams. So you convert gives you milligrams when you multiply it, and then you divide it by 1,000 to get grams. You get like 135 grams. So I wanted to know if that was accurate, and I searched really quick, and I saw there was a paper. I think it was from 1997. I don't remember who wrote it, but they estimated that there was actually one earlier than that, like the ICRP paper, that estimated that there's about 140 grams of total body potassium in somebody that weighs 70 kilograms. And then there was another paper written in 97 that said that on average men have about 1.9 grams per kilogram, grams of potassium per kilogram of body weight, and women have about 1.6 grams of potassium per kilogram of body weight. So if you did 1.9 times 70 kilograms, you get about 135, 136, somewhere in there. So these numbers are all working. This is all a sanity check. I can say to myself, I understand potassium homeostasis on normal physiology at this point, at least to the degree that I can make some predictions and calculations and be very, very close to accurate. And so next I'm going to go into hyper and hypokalemia. We'll talk about the specific etiologies and their mechanism.